So good afternoon. So we are to our last session, and the first speaker in the last session is Shikhar Patranabis. I hope I have pronounced the name okay. And he's a staff researcher in the foundational cryptography group at IBM Research, where he is working on theoretical and applied cryptography. He is basically, he studied at IIT Kharagpur, from where he did his PhD. And he has been working on this area, which he's going to talk to us about. He has several recognitions to his credit: IBM PhD Fellowship, Qualcomm Research Innovation Fellowship, and few more. So I invite Shikhar to give his talk.
Thank you. Thank you for the very kind introduction. And thank you to the Academy for the opportunity to present some of my work in front of such an august gathering. I am part of the Crypto Foundations team at IBM Research, which is spread across New York, Zurich, and Bangalore. And today I'll be giving a kind of a very high-level introduction to the area of post-quantum cryptography, which is something that I focus on. And I'll sort of try to walk you through some of the recent advances in terms of design of new post-quantum cryptographic algorithms, standardization in this space, and some of IBM's offerings in this area. I think I probably do not need to introduce the term cryptography. I think most of you would probably be aware of it if you are using WhatsApp to send a message, if you are using your credit card to make an online payment, if you are signing on to a particular website, or if you are dealing in cryptocurrency, you probably have heard of cryptography. It forms the building block for modern secure digital systems, and it provides a number of very important guarantees, which includes confidentiality, non-repudiation, integrity, and authentication. So cryptography has really advanced over the past 40 to 50 years, and it has a very, very large number of applications. But for today's presentation, I will just focus on the most basic application of cryptography, which is secure communication, right? So you have two entities, Alice and Bob, who want to communicate with each other. And unfortunately, they need to do this over an insecure channel in which the adversary can essentially view the packets of messages that they're sending to each other. And the goal here is to have a confidential and authenticated channel, which means that Alice and Bob should be able to exchange messages that only they can see. So in particular, this adversary should not be able to see. And this adversary should also not be able to pose, for example, as Bob to Alice or as Alice to Bob, right? So they know that they're actually talking to each other. So for example, if you are sending a WhatsApp message to your friend, you know that your friend is the one who is receiving the message, and you know that nobody else apart from your friend is actually able to read the message. And there are many examples of such insecure channels. The most popular one that we use every day is the internet, but there's also telecom networks, there's wireless LANs, and many of these. And the magic of cryptography essentially is that we can establish this secure channel between two parties in the absence of any such pre-established secure channel, right? So this really is, I would say, the cornerstone of modern cryptography, and it was sort of introduced by Diffie and Hellman back in the 1970s. So here in this slide, I sort of break down the way in which modern secure communication happens. So you have what we call asymmetric key cryptography, where you actually assume the existence of a costly secure channel. And then you have public key cryptography, where you have no such secure channel pre-established whatsoever. And the way in which modern communication works is a hybrid approach, where you first use public key cryptography to establish this secure channel, and after that you can use symmetric key cryptography for whatever messages you want to exchange. Okay, and the reason for this approach is that public key cryptography is typically very expensive, and so you only use it to establish a secure channel through which you exchange a very, very small key. That key could be as little as for example 16 bytes or 32 bytes of information. And after that, once you have shared that key, you can use it to exchange several gigabytes of information. And that is really, really not costly because you use symmetric key cryptography there. But unfortunately, this magic of public key cryptography, where you can establish a secure channel without having one in the first place, it does not come for free. Okay? And in particular, the motivation for today's talk is the fact that for public key cryptography to be realized, you actually need to make assumptions on the hardness of certain mathematical problems. Okay? In other words, you need to assume that there are mathematical problems that are computationally infeasible to solve within a reasonable amount of time. I'm not going to go into more details of that or of talking about what we mean by reasonable amount of time, but an example of such a problem is the factorization of the product of two very large prime numbers, right? So I give you the product of two very large prime numbers, and there is no classical algorithm that using today's classical computing facilities can solve this within what I call a reasonable amount of time. And this is an assumption that has been used as the basis of a large number of modern crypto systems which fall within the RSA family. There are similar other problems that we rely on. Unfortunately, once we bring quantum computing mechanisms and quantum algorithms into the picture, the hardness of these problems don't necessarily hold anymore, right? In particular, you may have heard of Shor's quantum algorithm, which is this very famous algorithm from the 90s, which is able to use quantum algorithms for period finding and in particular solve problems like factorization. What this essentially means is that once we have quantum computers that are able to run Shor's quantum algorithm, all of the cryptography that you use today in your day-to-day services would be broken. And the guarantees that I talked about in the previous slides would no longer hold, right? So you're talking about decades-old cryptography, which has been standardized and deployed massively, will stand completely broken once we have quantum algorithms running on scalable quantum computers. Now, you might ask, can we run them today? The answer is no. However, we have had significant improvements in quantum hardware. And very, very recently, we have also had breakthrough improvements to the algorithm itself, which reduces the capabilities that it requires from the hardware, right? And so what that means is that this confluence of improvements in algorithms and improvements in hardware could really shorten the timeline for when you might have a cryptographically relevant quantum computer capable of running Shor's algorithm. More importantly, the threats to these algorithms does not only start when you have a scalable quantum computer, it starts today, because adversaries can essentially do what we call as harvest then decrypt. So in the harvesting phase, they take all of the encrypted communication that is encrypted or digitally signed using quantum unsafe algorithms. And once they have a quantum computer, there's nothing stopping them from going all the way back and decrypting or forging all of that cryptographically secure or authenticated communication that they have harvested. And this really, really has implications across the entire digital stack. So this brings me to what we call as quantum safe or post-quantum cryptography, which basically refers to public key cryptographic algorithms that run on classical computers. So they don't require quantum hardware to run. However, they are plausibly resistant to attacks by both classical and quantum computers. And this is what forms really the core of our mission towards securing the world's digital infrastructure for the quantum era. I just want to mention briefly that public key cryptography or quantum safe public key cryptography is actually technically different from quantum cryptography or quantum key distribution or QKD, which many of you may be familiar with. In particular, as I mentioned, quantum safe cryptography runs on classical computers, unlike QKD, which actually requires very sophisticated quantum computing infrastructure. QKD is also kind of partial at the moment in the sense that it provides confidentiality but not authenticity. So you still need to use quantum safe cryptography, you want to add authentication. And our view for the foreseeable future is that QKD would probably be deployed and used for very, very specialized applications, probably find applications in the defense sector, for example, whereas quantum safe cryptography is expected to be more broadly used as a drop-in replacement for today's cryptography, right? For example, in the internet or other applications where classical cryptography is deployed today. Of course, there is a trade-off. Quantum key distribution is believed to be information theoretically secure, so it doesn't rely on mathematical assumptions, unlike public key cryptography. However, it has its own set of requirements which kind of distinguish it in terms of applications. Okay, so with that, let me quickly move on to what are the main solution components for quantum safe crypto. And we have worked at IBM across this entire spectrum. And I'm really involved in the first two columns here, which is the designing of new post-quantum cryptographic algorithms and processes for standardization of these algorithms for widespread deployment, right? And in particular, here I'll focus on the standardization process, which is being carried out by the National Institute of Standards and Technology in the US. It is not the only standardization process for post-quantum crypto, but it happens to be the most prominent one. There are other standardization processes being carried out by organizations like ISO, for example, and many different other countries have run their own standardization processes. This is followed by two other very important applications which are becoming important now. The first one is crypto discovery, which is if you have a piece of software that is using cryptography, how do you understand where exactly cryptography is being used? And how do you know that that cryptography is either quantum unsafe or quantum safe, right? And this is done using what we call as a cryptographic inventory or a bill of materials, abbreviated as CBOM. And once you have done that, then comes the question of migration, which is you want to take all your quantum unsafe cryptography and you want to migrate to cryptography that is plausibly post-quantum or quantum safe. And I think in one slide, I try to summarize the main technological innovations here in this space from IBM. In particular, we have had thought leadership for a significant period of time in the NIST standardization competition, which ran for almost 10 years and sort of concluded earlier this year, at least one phase of it, in terms of both public key encryption as well as digital signatures. And we are actually present in several of the standards that have been announced. And more importantly, we also have now offerings in the space of crypto discovery and migration in this three-stage capability that we call transform, assess, and discover, where we are actually working with our cloud software and consulting businesses to help clients migrate to post-quantum cryptography. Okay, so in the rest of this presentation, I will try to walk you through some of the main mathematical techniques that underlie the foundations of post-quantum cryptography, why we believe that they are plausibly secure against quantum algorithms, and what is the kind of standardization efforts that have happened around that. And I use this picture because in some sense, the world of post-quantum cryptography, it is held up by these atlases, which are these mathematical techniques, right? Because if these mathematical techniques cease to be quantum hard, then this world falls, right? And here you can see I have sort of tried to summarize some of the most prominent techniques for post-quantum cryptography. And I'll spend a couple of slides just talking about this particular technique, which is called lattice-based cryptography, because this has been studied for the longest period of time in theoretical computer science, subsequently in cryptography for the past 40 to 50 years, and is also most prominent today in terms of the standardized algorithms, right? This is the technique that is mainly used. And this really relies on the hardness of two problems, which are called small integer solutions and learning with errors, for which we believe there aren't quantum algorithms. And I really won't have time to go into the details of these, but I just want to mention that they are essentially geometric problems, okay? Unlike a problem like factorization, which is an algorithmic number theory problem. And this is the main reason why Shor's algorithm does not really extend to these kinds of problems. So here in this slide, I give you a representation of a lattice, which is like this matrix or a grid. And you can think that each intersection of two lines here is a point on the lattice. And then you can think of some problems in this space. For example, suppose I give you this grid, and I ask you to find the shortest vector in this lattice, which is basically if I have an origin here, then I ask you, for example, let's say this is the origin, I ask you which is the point here which is closest to the origin, right? Similarly, I can give you a point that is not exactly on this grid, but maybe close to it. So you can see this green point is on the grid and this red point is close to it. And I ask you, can you give me the point on the lattice or on the grid which is closest to this point? So these are the kinds of problems that actually Shor's algorithm is not able to solve because they are very, very different from classical problems such as factorization or discrete log, which is a related problem. And these classical problems, they typically reduce to what we call as a broader problem called the hidden subgroup problem, which can be solved by Shor's using period finding. Okay? And it doesn't really seem to work for these kinds of lattice problems. Another way of interpreting these problems is that they're actually machine learning problems. You're trying to learn some secret, but unfortunately the instances that are given to you are noisy. And it turns out we don't have good quantum algorithms for denoising these problems for particular choices of parameters. Okay, so that's, if you are interested, if you come from a machine learning side of things, then maybe this interpretation would make more sense. And then there are some other techniques that have also come up in more recent years. These are in different stages of being analyzed by the community, and so we have different levels of confidence in their hardness. I really won't really go into the details. So this is sort of the broad timeline, I would say, for quantum safety. So the NIST PQC competition in particular, it sort of ran for the first set of standards were announced in 2024, but it really in earnest finished its first phase in 2026, and we still have some processes ongoing. In particular, the main project ran for 10 years. It had submissions from all over the world, and eventually it standardized 2 public key encryption algorithms and 3 digital signature algorithms. And among them, Crystals Kyber, Crystals Dilithium, Falcon, and Sphinx have all been led or co-led by researchers from IBM. And then we now have a new call for submissions, which started in 2023, which is for additional digital signatures. And this was, the round 3 of this was announced earlier this year. And we have 4 out of 9 submissions here from IBM, and I am involved in this one called SkiSign. The goal of this additional standardization is that, as I mentioned earlier, lattices feature very prominently in the NIST standardization process. However, if lattices were to break tomorrow, which could happen, then it's sort of like putting all your eggs in one basket, and we don't want to do that. So we want to diversify the portfolio of assumptions from which we know how to do cryptography. It's almost like diversifying your investment portfolio, if that makes sense. And here's a sort of a comparison of the 4 algorithms from IBM, which are currently in this additional standardization process. And I just want to maybe focus on this part of the slide. So the fact that we have the classical standardized digital signatures on the left. So these are widely used, widely deployed today. This one is based on RSA, and this one is based on elliptic curved. It's an elliptic curved digital signature algorithm, or ECDSA. And you can see that the standards that we have today, the post-quantum standards, they're really inefficient. They have much larger signatures and key sizes. And so now the goal of this additional process is to come up with alternatives which would perhaps be closer to these in terms of the compactness in particular. And this is achieved by a subset of these, right? In the interest of time, I'll keep the discussion at this level, but I'm happy to talk offline about this. And in particular, I'm involved in the design of SkiSign, which is currently in the 3rd round of the NIST process. This is joint work with a lot of researchers globally, but also with my wonderful colleagues Andrea, Luca, and Basil from IBM Zurich. And here we actually have significantly smaller signature sizes and smaller key sizes than the lattice-based standards that have been announced. Okay. And SkiSign is really, it shines in applications where you care about having very compact signatures and very compact keys. And this is really important because you may have applications where you need to store these keys in the longer term, and optimizing for storage is important. It's also important because the signatures being smaller means that they require less bandwidth to transfer over an insecure network. And there are more details available. And there's both ongoing standardization as well as a lot of cutting edge research happening in this space. It's a very exciting area in general. Okay, so I want to spend the last couple of minutes talking about, suppose you are an enterprise or an organization and you are looking to adopt quantum safe cryptography, right, going forward, then how should you go about it? Now, there is a very fundamental principle in cryptography, which was introduced by the Danish cryptographer Kirchhoff in the 1850s, which was that a crypto system security should always be based on the secrecy of the key and not the secrecy of the algorithm. You do not want to use your own proprietary algorithms for doing cryptography. You want to use algorithms that are publicly known, that have been standardized, that have been cryptanalyzed over a long time. And all you want to do is you want to make sure that you are keeping the secret secure, right? So this is particularly relevant because in many cases, people are hesitant to use NIST standardized algorithms because they feel that that NIST, being an US agency, might introduce backdoors. The point is that these algorithms are only standardized by NIST. They are not proposed by NIST. They are designed by the wider community, which involves a lot of cryptographers from many different countries. And they have been studied for a long, long time. So there aren't any obvious trapdoors. However, it can happen that some adversary could subvert the security if you have an incorrect implementation of this. So it's very important to implement your cryptography securely. And that's another whole talk, which I don't have time to go into. But as long as you are implementing your algorithms correctly, you should not have any issues in adopting these algorithms. And this is the principle on which, at IBM, we are helping our clients and partners prepare for quantum safe future with what we call as crypto agility, which is you are agile enough to be able to migrate from quantum unsafe to quantum safe cryptography. There are products and services which can help you do that, in particular with IBM Z, which is our cloud business, and IBM Guardium, which is our main offering in the quantum safe crypto space. There are demos available for these online, so if you're interested, please do check them out. And finally, of course, being from research, I'm interested in standardization and community, having community impact, and we are involved in a number of these. The most prominent one being the Linux Foundation Post-Quantum Cryptography Alliance, which was launched about 2.5 years back. And we are a prominent player in this space because we have been working with Linux Foundation on standardizing and helping people migrate to quantum safe crypto. That's all I have. Thank you, and I'm happy to take questions.
So questions?
So my question is that are all the PQC algorithms based on the not being vulnerable to Shor, or there are various other attacks that you are looking at?
Yeah. So most of the algorithms that I presented in the earlier slides, there are no known quantum algorithms for them. I think the main known algorithm for solving them is, there are algorithms that solve subgraph isomorphism or hidden subgraphs isomorphism, right? So there haven't really been any algorithms that can solve them better than sub-exponential time at the moment. So Shor's obviously does not work, and then we don't have candidates for other algorithms, although this is a very active area of research and people are looking at potential algorithms for solving them, right? So one thing I do want to mention is, if you are from a computational complexity background, none of these problems are NP hard, right? Which means that, we don't have a huge amount of confidence in them. We just know there aren't algorithms at the moment to solve them.
Any other?
At a very high level, can you tell what is this isogeny-based approach? I do not know this term.
Yeah, so the isogeny-based approach, it actually relies on the fact that there are these algebraic maps between elliptic curves. And the problem is given two random elliptic curves, compute the isogeny map between them. And this is a problem that we believe is, well, at least at the moment, there aren't any algorithms that do better than sub-exponential. And there are no known quantum algorithms that do significantly better.
Hi, sir. Yes, sir. My question was, this PQC is making the problems difficult to solve, right? So how fast do you expect the hackers to catch up to it? Because say if I intercept the asymmetric key while it is getting transmitted, I can, and after that the symmetric pathway is established, I can then get all the data.
Yeah, so that is exactly the threat model. But the idea is that these PQC algorithms, they provide you a mathematical guarantee that as long as you only see the messages exchanged publicly between the two parties, Alice and Bob, you cannot actually find the key that they have shared, right? So this is the hardness problem. And the conjecture is that there aren't, for example, in these cases, quantum algorithms to solve them. Or in the case of factorization or discrete log, there aren't classical algorithms that can solve them.
Sir, as you told that for the case of the cryptography, so as you told, actually the key distribution, which is the key is the main factor than the algorithms, right? So my question is, how many number of qubits which are basically needed for say generating the key? So minimum number of qubits, and up to what extent the current level of work is going on?
Okay. So first of all, I think this is more relevant to the quantum key distribution that I mentioned, which is different from post-quantum cryptography, because post-quantum cryptography doesn't require qubits. It just runs on plain classical computers. I think for QKD, I'm not really a quantum expert, so I won't talk about that. But what I'll mention is that currently we are able to exchange only very few qubits, for example, right? And that may be okay for some very limited applications, but it requires very specialized infrastructure, right? And I think there have been some toy level demonstrations all around the world, including in India, but there's still a long way to go before we can actually start deploying it at a wide scale.
And one more, sir, for hard level problems, which among these, which is the better one?
It's hard to compare, right? So for example, if you talk about the community's confidence, the highest is in lattice-based questions, because those have been studied for a long time, also in the theoretical crypto literature, because they are geometric problems. But I would say that they offer you different trade-offs in terms of performance and efficiency, right? So that is the main reason why we are looking at a number of these alternatives. The other reason is to diversify, right? So yeah, so some of these probably we believe are harder than others, but it really depends on the level of research that somebody may be able to find an algorithm to solve them tomorrow. We cannot rule that out, basically.
Okay, thank you.
I think we'll stop here and we'll move on. Thank you, Shikhar, for a very nice talk. So our next speaker is Dr. Santosh Chauhan. Santosh Chauhan is from CCMB, Hyderabad, and he did his PhD at AIMS, Delhi. And after doing his postdoc, he joined ILS Bhubaneswar and then moved from there to CCMB, Hyderabad. He is going to talk to us on something very interesting. We all have heard about programmed cell death, but he is talking about programmed cell revival. So let's hear the interesting talk. Santosh.
Thank you, Dr. Batra. Thanks, Dr. Raghavan, for this opportunity, and the Indian Academy of Sciences for this opportunity. I'm a fellow from 2023. This is my first talk in this kind of conference. So basically, because I know there is a varied expertise here, so I'll keep it, this is a complex study, but I'll try to keep it under control. So I will talk inside the cells. So everything is inside the cells of our body. So please keep that in mind. So my lab work on cell biology, and in cell biology also, we are more interested in innate immune responses of our body and other aspects like autophagy and other, which I'm not going to talk today. I'm going to talk about one of our recent work published, which is programmed cell revival. Many of you have heard about programmed cell death during your, maybe in bachelor's or before bachelor's. Programmed cell death is, what is it? It is most of the cells which are dying in our body are very well programmed, means they are dying by a programmed process. And this is very well established from years that how they actually go towards the death. And programmed cell death is very important in every day in our body. Millions of cells might be dying by programmed cell death, and it is important to maintain, to keep us away from many diseases, including cancer and autoimmunity. So what is this programmed cell revival? Okay, so I would like to convince in like 10-15 minutes that what doesn't kill you will make you probably more stronger, at least at the cell level. Okay, so this paper is published in AMBO Journal, and this was present on the cover where you can see the phoenix rising, and that's what it depicts from our study. So this study was not actually distant. It was totally observation-based study and totally a serendipitous observation. We were studying something totally different. We were studying the cell death, actually. And this guy, Kautilya, he was my first PhD student, and he wanted to understand the cell death, some aspects of cell death, and specifically lysosomal cell death. That's, if you trigger the lysosomes, you know all lysosomes probably, there's a part of cells which is like a suicidal bag. If they burst, they will kill the cell. So here these are all agents which can kill the cell by lysosomal burst. Okay, so I'm keeping it all these things simpler. So what he saw is, what he was trying to see is how lysosomal cell death happens. What happened is he went to the lab, he started this experiment to understand, and so what happens is, this is when you give these agents, the cells get these morphology, means they are towards all started blabbing and they come out of the surface, which is the property of these cells. These are fibroblast cells. They come out of the cells into the media. And this is the point where this guy has to collect the cells because we want to study at this stage. Luckily, sometimes these things are luckily, he forgot it to collect these cells. And when he went after around 1 hour, because he forgot it, this come first. These cells are like, standard apoptotic cells in literature. And most of the people at these concentration collects these cells. There are hundreds of papers on LLOME. So they all collect these cells at these stages. So you can see that this is faster. So I will go with this. So they completely get detached. They have all apoptotic things going on, and then they revive back. Okay, so those also revive back. Or different magnification these cells are. Okay, so we try to understand. So what we did is we try to understand whether what we were using this 4 millimolar concentration, which is the standard concentration across the field, and we thought that something is wrong because it is not possible the cells can revive from complete cell death if it is the truth. So we did concentration curve, and you can see that these 4 millimolar are still better than 8 millimolar. But you see the 8 millimolar, nobody can predict that these cells going to revive, but they will revive. Okay, so they revived. All of them, most of them. Here you will see all of them. There you see most of them. And 12 millimolar, you will see none of them. So we found that this is a sublethal concentration which people were using, and it's actually wrong to use it. But it gives me another thought process to follow. So, and we found was that, okay, there is one more video. What we found was that cell death, what people use in the literature, is by these annexin FITC. If the membrane of the cell gets these annexin FITC positive, they take it as a cell death. But that's completely wrong. Until the nuclear membrane, the nucleus inside the cell, until this disrupts, there is no cell death. They can revive back. That's what we showed in the paper, and this could be a redefinition of proper redefinition of cell death. And you can see here that these cells revive back because they have not taken this stain into the nucleus. Once they take a stain inside the nucleus, because of the nuclear membrane disruption, they cannot revive back. So we defined it. We also defined how the cell organelles, different kinds of cell organelles we tested, check that inside the microscope. These are inside the cells. These are different organelles. You all know probably the tubulins, mitochondria, Golgi, and some complicated ones, autophagosome, endosome, lysosomes. We checked that at this stage when the cells are going towards the death, they all get disrupted completely because these are floating cells. They are all getting disrupted. So from there, coming back to the normal life is very interesting for us in terms of cell biology, in terms of molecular biology, in terms of every biology process. It could be the possibles. So till now it was cell revival. Then we gone into the molecules. You want to understand what is happening, how these cells are reviving back. So this stage is mostly what I was showing. They come up, and these are all stages when they revive back to the normal. So we did the transcriptome to understand the level of gene inductions or mRNA induction or the to understand what is happening here, to be very easy on that. And what was very interesting will come from this heat map, that until the cell are dying, this was the C3 stage where the cells are in the supernatant and have all the properties of cell death. When the cell revive back, they revive back very interestingly. There are clusters of genes which comes up. And these are not 1 or 2 genes or 100 genes, or these are 2,000 genes. These are around similar 2,000 genes. So it's very well programmed to revive back the cells into a life. And when they revive back, I'll not again take you what these cluster 3, cluster 2, cluster 1 are really. To be precise, what was happening is they are coming back to life, something like they know that what was then, they were like a embryonic developmental stages, they know that what they were initially. So when you see this cluster 3, the pathways which were activated, means the genes which were activated, is developmental signaling was getting activated. Almost everything you see, that all circadian rhythm, embryonic organ morphogenesis, cell development, muscular structure development. So these cells somehow are like, they have cleared what they were, and they are restarting their life. And you can see that what goes next is metabolism, nucleotide synthesis, protein synthesis, everything was kicking in a very programmed fashion. So that's the reason we called it programmed cell revival. And if you Google that programmed cell revival, you can actually see more detail if you are a biologist and interested in it. Because of time, I may not go in that detail here. And for the biologist, what kind of pathway it was we found is, you can see all the pathways like Wnt signaling, Notch signaling, Hox signaling, DGF-beta signaling for the biologist. We found it, the first cluster 3 has all those things, and then you have metabolic pathways, you have inflammatory pathways, all these required for the revival of these cells. Okay, so again, this is quite a complicated. Just want to tell you that when the cells are reviving, all these gene signatures are not random. They take some kind of program. They go up, go down. Mostly they go up as they revive back. Okay, what's there? There was a transcription signature. We thought might be that's just not a transcription signature, but there will be a genomic signature also. So we start looking at the epigenetic level. So not going again in the detail, what we found it that at this stages, when the cells was in a completely apoptotic stage, and when they are reviving back, the cells have lost their epigenetic informations. So this is the ataxic data, and you can see a lot of open chromatin at lot of genes, which means that when cells lost that, they lost their differentiated state, and they have partially acquired a de-differentiated state, which is more like a developmental cells or initial cells. So that's what chromatin opening data tells us, which leads to higher transcription at multiple loci. So that was the thing. This is all about LLOME being used to study how cell go to sublethal concentration of LLOME leads towards cell death. But if you actually give sublethal concentration, they can revive back. And when they revive back, they are like new kind of cells, which is more or less using or they slough off all the information, and they are reviving like they are in the coming to the new life. What was we thought of is that point of time that these all molecular pathways and looks like they are similar to what happens during wound healing, tissue regeneration, and all embryonic growth. So they have a lot of similarity between these three processes. I can tell you there are tons of literature that wound healing, when the wound heals, they are also having similar pathways, which we are seeing, even during the tissue regeneration. For example, you have tissue regeneration of in different models, tail regeneration, or any kind of, these pathways are getting activated. Developmental pathways comes first. So we thought that let's check it out and whether this LLOME can actually cause some kind of interesting stuff in terms of wound healing, for example. First, we have done a lot of things now. This all work is filed for patent for India in 8 different countries now. So what we found was that this is name is changed, but this is LLOME. So what we found was that by the 3-day, this is a wound healing model where you can cause a wound on the mouse and you can actually look for the different days, how fast it heals in multiple mouse. So one of the mouse we are showing, this is wounds which are made in the first day. And you can see that by the 3rd day, when the wounds are still fresh in the untreated mice, you have almost closed wounds with the LLOME treated topical treatment of the LLOME. We have another model with our collaborator where you can actually, it's called, you can injure the epithelia of eye with NaOH and scraping. And there also he saw the same thing, that if you treat with the LLOME, there was much faster recovery, much faster recovery. Within 1 or 2 days, the reepithelialization was taking place. There is another collaborator who was working on frog tadpoles tail regeneration model. There you can add LLOME directly into the water of these tail cut tadpoles. And when we put that, as you increase the concentration, there was much faster, after the amputation, much faster tail growth. There is another collaborator who works in ACTRAC and also NBRC. So we try to understand whether the regeneration in C. elegans model, the axon regeneration in C. elegans models can be faster, and he found regrowth length as well as recovery index, which is this. These get recovered faster by the when they dissect it and they cut this axon. First of all, regeneration was faster, and also the recovery of these C. elegans was faster. In another model, Drosophila, where they found that very interesting, that the blood progenitor indexes, hematopoietic progenitor stem cells were increased in many folds in blood lymph glands when they are fed with the LLOME. So this is really interesting to us. We are trying to understand many things right now, and we are taking these different models. Like this model also we are taking into the mouse model, which is now funded by ICMR by Center for Advanced Research grant. We are taking that model to understand, can we induce pluripotent stem cells faster? We are actually trying to understand whether we can actually heal the diabetic wound models faster. We are taking this to rabbit and probably to the human in this Center for Advanced Research grant. So with that, we start with the observation. We end up with multiple PCTs, multiple grants, and also beautiful basic work. These are all people who have collaborated, including IASC and multiple institutes all over the country. And this was the paper published last year. And this is my lab, and financial support with different agencies, highly thankful to them. Thank you very much.
So questions, Partho.
One very quick question. Towards the beginning of your talk, you showed that as you increase the concentration, the number of cells reviving decreases. Later, what you show is as you increase the concentration, the speed of recovery increases. So is there a sort of opposite?
No. So the thing is, those are cells. These are whole body models. They are very resistant. They don't care much about that concentration. So cells are, because we are putting directly onto cell culture, that's what the dynamic is. That's what the concentration gradients were. But in human, so in the mouse, we found even 20 millimolar we are giving from last 6 months, they are fine. So this different is because of different whole body models are very different from there. So there is no lethality by now we have seen with these concentration which we are using in the cell culture, to be precise.
Just one quick question. So have you looked whether in the context of tumors, whether these enhance tumor growth?
Very good question. So that's what we are looking for, what we feel, to be precise. In cancer cells, because I tell you what's going on right now in this field. This is, in the cancer cells, when we give all these anti-cancer drugs, our hypothesis is most of these getting into the stem cells is because of this process. Because you give so much of them, they are not going towards fully death, and they recover back with much potency, probably by programmed cell revival. So in the context of cancer, this could be a very bad process because it allows these cancer cells with the resistance of equivalent to the stem cells to bounce back much faster with much probably potency as compared to the if they revive, if they don't die or they revive without going this process, they revive or something like that. Yeah.
So 2 questions. First, when you look at different cell lines and different tissues, are the transcriptomic signatures at different time points similar or do they vary?
So tissues we have not done. Now we are doing in wound healing model, which we're gonna go for time points. But in cell culture, it is absolutely same. We have done it with 2-3 different cell lines.
The second question, compared to an acute injury, when you look at, say, diabetic ulcer you are trying to think of, do you think the mechanism is going to be similar or?
So I think you're absolutely correct when you are seeing the cells versus you are seeing a model which is whole organism model, the many mechanism may kick in. And here also I'm not claiming that I have found all the mechanisms. Because these cells who are going towards it, they will secrete a lot of EVs and a lot of things which may actually call upon the immune cells. There will be a lot of things going on inside the organism. So in the inside organism, this mechanism may lead to, for example, macrophages, coming to macrophages inside this environment and clearing of the debris. And maybe the immune cells get activated. So inside the organism, maybe this mechanism may leading to many other kind of responses.
Good afternoon, sir. I was just wondering if, yesterday we had a talk by Professor Jagadish, and he had a shockwave way of healing wounds. I was just wondering if there is a way to apply shockwave first and then the pluripotent stem cells from LA1 can be added. In fact, you have some collaboration with the AAC. Maybe next time, this is something that we could look at.
So Shockwave may be doing the similar. See, with this thing, what we have done is we have given a kind of mechanism to the world that may not be LLOME or lysosomal cell death driven. What we are exactly saying is, if cells are going towards, so Shockwave may cause similar kind of programmed cell revival, and they may induce similar kind of stemness. So that's maybe one way of doing the programmed cell revival for therapies.
Good afternoon, sir. I have a question. Like, in context of humans, do you think any cells specifically, or like any types of tissues are susceptible to cell renewal, and why do you think so? And are there any biomarkers to detect this cell revival?
Very good question. The first one is, we mostly tested these cells in these things in fibroblast cells and epithelial cells. But if you go towards terminally differentiated cells, such as your immune cells, the U-turn is not possible by the day because we have tested it. So the terminally differentiated immune cells don't take a U-turn. So you have to have these fibroblast cells in your environment, in tissue environment, and epithelial cells, which is always the case. But terminally differentiated immune cells doesn't take a U-turn. What was your second question?
Are there any biomarkers to detect this cell renewal?
Biomarkers to check the cell. That's a good question. I don't know. We didn't try it.
One more. Like let's say the stress event that caused the cell to go to apoptosis or trigger the cell death, does it have like epigenetic memory? And if it does, like, you know, of the stress event, and when it gets like revived or gets under the process of renewal, right? So does it like the cell is becoming more aged? Does it age more like, you know, fast, or like does it like have the memory of the stress conditions?
So what we found, it ages slow. In Drosophila model, what we have already checked, it's if you feed the larvae with the LLME, the aging has, and this is patented also again, the aging is getting suppressed. Now we are going for animal model. Just I told you that we are checking that in animal model, and we have injecting them with that. So that's aging is slowed down actually.
So I'll ask the last question.
Sure, sir.
So what about in vivo? So in vivo cell revival, any incidents?
So we have checked in this paper to some extent. We cannot actually, we have done multiple FACS studies, multiple other ways of doing, taking the tissue from
without the injury I'm talking about, to spontaneous.
Oh yeah, I'm sure that is happening.
No, but nobody has
means injury, this is injury lead. Wherever the injuries are, I'm sure this is happening.
No, injury is happening, but
normally our body all the time goes through the injuries.
Death.
For example, liver. We are injured by multiple dams. Our liver is one of the places where tons of dams are being accumulated and getting the injury. I'm not talking about the physical injuries. Our body normally responds, gives a lot of injuries.
Right.
And they're inducing the stemness will always,
so
stemness or dedifferentiation.
Revival is maybe happening, but you have to show that, right?
I can predict it.
Yeah, okay. But another curious thing is that whenever somebody is studying apoptosis, they do this dose-dependent things. So how was it missed earlier that lower doses are?
I don't know, but the 12 millimolar, I don't think many people goes up to that because they see everything by 4 millimolar. When they collect the cells, they go off. This is by chance we didn't collect the cells.
Okay.
So mostly 4 millimolar people go, 8 millimolar may people go. Okay. There also revival is there. But 12 millimolar, I don't think someone has tried. It's just death in 5 minutes.
Okay. All right. So we'll stop here.
Thank you.
Thank Santosh for a very, very interesting. So our last speaker is Akshit Goyal. Akshit Goyal is a faculty member in biological physics at the International Center for Theoretical Sciences, where he is working on understanding the collective dynamics of evolving ecosystems. And today he is talking to us about towards a theory of evaluation of ecosystem. And he earned his PhD from NCBS and has got some recognitions, and currently whatever he's on to, he's going to talk to us about. Akshit.
Thanks. Can people hear me in the back? Okay, great. Thanks a lot for giving me an opportunity to talk. So I am indeed going to talk today about our efforts to build a theory for the evolution of ecosystems, and I'm going to describe what this even could mean. But I'll just start off by just reminding us that all around us, we see lots of different kinds of ecosystems, and a common thing to many, many different kinds of these ecosystems is that they maintain just an incredible amount of biodiversity. And so in all of these ecosystems, there's many, many different species, and so they all interact with each other, they self-organize, and through these kinds of dynamics thrive. And I would say that, of course, we know that this happens, but really if we want to understand quantitatively what is happening to the dynamics of these ecosystems, I would argue that in sort of the recent years, it's really in the microbial realm that we've been able to get unprecedented quantification of the dynamics of such systems. And so just very, very quickly, what these techniques involve is that you go out to some sort of an environment that you care about. You might care about the soil. You go and you scoop out a sample. You scoop out many, many samples, and you send it down through some DNA sequencing machine. I won't describe the details of that. You do some sort of analysis, and that analysis tells you what kinds of species there are, how many. If you do this over time, you can get the dynamics. And not only can you do this for soils, you can do this for marine ecosystems, you can do this for the gut, you can do this for different kinds of lab ecosystems if you want. And one thing that we've learned by doing this again is just like the macrobial ecosystems like forests and the oceans, it's also true in the microbial realm, it's even more so true that there are just an incredible amount of biodiversity. So just to give you some numbers, in every gram of soil, you can see between 1,000 to 10,000 species, between 10 million and 1 billion cells of different microbes per gram of soil. These are just crazy numbers. And so because of the recognition of these crazy numbers, what we've also realized is that the classical paradigm of understanding ecology and sort of ecological dynamics, which was built on this intuition of low diversity, built on saying that if we understand what happens to 1 or 2 species, we can understand what happens to more, simply doesn't work anymore. And so in the recent years, what people, including us, have been able to come up with is to interrogate what happens to highly diverse ecosystems directly. And so just like in these previous kinds of work, what was really important is to think about these systems as systems that we can study using the methods of nonlinear dynamics. What is important or what has become important are different mathematical techniques, particularly from statistical physics and random matrix theory, that have let us probe what happens to high diversity ecosystems directly. And if this seems kind of mysterious, let me just very quickly tell you what kind of thing inspires us. So a very different problem, which was facing people in the 50s and indeed the 40s, was to understand the uranium nucleus. For various reasons, people wanted to understand it. And so, this was really problematic because the uranium nucleus is very, very complex. It has, 92 protons, 138 neutrons, and so on and so forth. And so, this was a very, very hard problem to solve. And so, Wigner over here came up with this basically genius move, which is that he said, okay, the uranium nucleus is really complex. I don't know how exactly to model it. What I'm going to do is that I'm going to assume that this complexity just means that I can replace the properties of the uranium nucleus with some random matrix, okay? And this had no right to work, but what Wigner and since then others showed was that this theory that was based on imagining the uranium nucleus and its couplings as a random matrix did pretty well, especially well to predict one of the problems that people were interested in at the time was, which was to predict the energy level spacings of the uranium nucleus. So, the details are not important here, but you can sort of figure out what are all the different energy levels of the uranium nucleus. And in particular, you can ask for the spacings or the differences or the gaps between these different energy levels. And so here is, for instance, what that distribution of these energy levels looks like. Over here in this histogram in the back is data. And you can see in this curve over here, this is a random matrix prediction that came from Wigner. You can see it's a pretty good match. We understand many details since then of why this works well, et cetera, et cetera. And so what we've been trying to do over the past 5 or 10 years is to import these kinds of ideas into studying ecological systems. Okay, and so just to give you a very quick caricature of how we are able to do this, what we imagine is that we imagine models where there are species, there are many, many species, and there are different resources, different kinds of metabolites that these species need to consume. And what you write down are a large number of systems of equations, both which model the dynamics of the species and those that model the dynamics of these resources. And these two equations, these two sets of equations are coupled to each other, and they contain some pretty simple processes. For the resources, of course, the resources are supplied at some rate. They're depleted by the different species around them. That's how species grow. And of course, it might also be possible, which is especially true in the microbial realm, is that different species, when they consume resources, are also able to secrete other things as as sort of metabolic byproducts. This feature is called cross-feeding, and so you can put that in as well. And so different species will grow by consuming resources, and then they might be paying some sort of maintenance cost to be able to work with these, right? So the details of the models are not so important. The important thing I want you to take away is that there are really a very large number of species, which means that this species equations are not one equation. There's a large number of equations, one for every species. For resources, again, there's not one resource. There are many different resources. And so there's a large number of equations for the resources. And typically, when we say highly diverse in statistical physics, what we mean is that we're interested in a limit where the number of species and the number of resources go to infinity. And so for those of you who are not used to this notion, it's actually true that when these numbers go to infinity, things become easier to solve, et cetera, et cetera. But the important thing that comes up when you're trying to write down these models is, just like sort of Wigner was sort of worried, how do you figure out and choose what all the different parameters are? For instance, the parameters in these models could be parameters that have to do with which species consumes which metabolite, which species produces which metabolite. We don't have access to all of this information. And so what we do is borrow a trick from Wigner from the 50s, which is just like Wigner was dealing with the complex uranium nucleus and replaced that complexity with randomness. We take the complexity of metabolism, the complexity of these different ecological dynamics, and we replace it with a random metabolic matrix. Okay, and so by doing this, what we've been able to show over the last few years is that this can help us describe the statistical properties of the dynamics of these ecosystems, just like Wigner was describing the statistical properties of the uranium nucleus energy level spacings at different scales of ecological complexity. So we've shown that this can work to describe the dynamics of ecosystems that you bring to the lab, so like simple laboratory microcosms, but also to describe the dynamics and properties of different gut microbiomes. These are microbiomes that we have in our gut that help us digest our food, but then all the way up to marine and planetary scale ecosystems. Okay, my interest here today is, usually I would have given you a talk about one of these things, but my interest here today is not to give you a talk about one of these things, but to give you a talk about what is not in any of these models, which is a big, big caveat, which is that all of these models ignore the basic evolutionary process that has created and sustained the diversity of these species. Okay, and so this is actually a big, big challenge. And the challenge really is not to understand how the ecology of many species works, but instead to understand how ecology and evolution work together. And the reason this has been a big challenge in the field of ecology and evolution is been that these two processes have usually been studied separately. If you go to an ecology and evolution department in the US, they are one department, but there are ecologists who work studying ecological dynamics who pretend evolution doesn't exist. There are evolutionary biologists who pretend ecology doesn't exist. This is a simple caricature, but this is not far from the truth in my experience. And so really the reason that this is the case is not because people are stupid, it's because actually this is a very challenging theoretical problem. We don't know what we should expect when the processes of ecology and evolution run together. And so, I'm not claiming that we solved this problem, but I want to tell you in the second half of my talk about some recent work that we've been doing to take the first steps towards trying to incorporate evolution into ecological models. Okay, and so what I'm going to tell you about is we're going to play a simple game. We're going to start with a simple evolving population. I'm going to tell you about what the standard evolutionary paradigm of population genetics is. And we're going to ask what happens to this paradigm when you add a diverse ecological community of the kind that I was just telling you about. Okay, and so before I do that, I have to tell you at least something about the simple evolutionary paradigm. So the classic paradigm, the paradigm of population genetics, asks many questions. One of the simple questions, the classic cornerstone of the field, is you start off with a population of many, many individuals. Let's imagine that all these individuals are what we'll call wild type individuals. And into this population, you introduce a new individual, which is a mutant. So you can imagine this mutant comes up from one of the wild type individuals that reproduces and mutates. And one simple question that you might ask yourself is that if I wait a long time, what is going to be the fate of this mutant? And there are really traditionally two answers. One is you wait a long time and this mutant goes extinct. You go back to the same picture that you were with. It's only wild types remaining. Or you wait a long time and this mutant does what is called fixes. It fixes in the population. Now all you see in the population are just these mutant individuals. You don't see wild types anymore, right? And so in some sense, when you ask the question, what is the fate of the mutant, you're asking what's the probability that this mutant goes extinct versus fixes, okay? And the classic way that people have tried to answer this question is to realize that these mutants have stochastic dynamics, which is to say that if I plot the frequency of this mutant, which is the fraction of the mutant individuals in the population, and I track it over time, there are many, many realizations that might happen because these dynamics are stochastic. There's birth and death events, which are random, and the randomness of these birth and death events results in some trajectories of mutants that go up all the way and fix, and some other trajectories that don't go up all the way that go crashing, and when you get frequency 0, the mutant's gone extinct. And the classic idea, and of course the mutant starts off here at time 0 at some very low frequency, usually one individual in the entire population. And so usually the classic way of sort of describing what happens to these dynamics mathematically is to solve what is called the Kimura equation. I don't want to go into the details of the equation, but the idea really is that this equation captures these two processes, one of selection, which just says, okay, the mutant grows better than the parent by some amount, which you call S, which is a selection coefficient. And of course, this process is not deterministic. There's a stochastic component. There's a noisy component which captures the stochasticity of the birth and death process. And so one of the things that we learned by solving this equation and by sort of studying it, this is sort of classical work done by Kimura in the 50s, is that there are really two different regimes. At low frequency, when the mutant is very, very rare in the population, stochasticity dominates. It's really the stochastic part of this, of the dynamics of the mutant that matter. But if the mutant manages to get up to some critical frequency, then actually selection dominates. And if this mutant happens to be fitter than its parent, it's almost guaranteed to fix in the sort of population. And so you can solve this equation and ask and answer what is the fixation probability of the mutant. Okay, and so the dogma sort of classically in the field has been that this equation and this process, which contains only the parent and the mutant, that is what was there in this picture that I showed you, describes very well a very wide range of biological situations. And so what I want to tell you and what I want to remind you is that this is very different from the picture that is going on in nature. The picture that's going on in nature is not that there's just the parent and the mutant and they're slugging it out. It's that the parent and the mutant are actually embedded in a diverse ecological community. And so if we want to study the processes of population genetics, we ought to do it in this context, embedded in these diverse ecological communities. And so that's exactly what we did, which is that we asked, can we write down and describe a very general theory of what would happen to the parent and the mutant when they're embedded in this big community soup? And so really, what I mean when I say general is that we're going to imagine, again, that there's a large system of equations we need to solve, one for every species in this community. And these equations have two components. One is the component by which these species or strains grow. And the other, this component of noise, which captures the stochasticity of the birth and death process that I was telling you about. And what we have and what we've been able to do is to come up with, in statistical physics language, what is called an effective theory of the dynamics of this mutant embedded in a diverse community. And when I say that this effective theory is universal, what I mean is that this G sub I, this growth function, how these different species grow, can be your favorite ecological model. So it can be any model. And so it can indeed also be a complicated model of the kind that I was showing you a few slides ago, where there are species, there are resources, species compete for resources, etc., etc. And you can imagine that there's one equation for all these different species in the sort of community. There's also these kinds of equations, one for the parent and one for the mutant. And you can imagine that the parent and mutant are similar to each other by some metric that you might devise. And our goal is going to be to predict the dynamics of the frequency of this mutant in the soup of this community. And so when I say the frequency of the mutant, I mean what fraction of the sum of the parent and mutant population is the mutant. So if this frequency is 0, everything is the parent. If this frequency is 1, everything is the mutant. If this frequency is 0.5, it's half mutant, half parent. And so our goal is really to write down some sort of equation and study the dynamics of what this mutant's frequency does over time, just like the analog of the Kimura equation, which didn't have any ecology. Okay, and so my claim is that we can do this, and we can do this with a technique which is called dynamical mean field theory in statistical physics. I won't belabor you with what the details of that theory are and how we go about doing this, but I'll just tell you what the punchline is. And the punchline is that despite the ecological complexity of the community, despite the fact that there are a very large number of species, what you find is that compared to the Kimura equation, which just had selection and some effective population size, you need only one additional parameter to capture the complex feedbacks due to the community and their effect on the mutant. And so what I mean by that is that you can take this entire picture where there's a mutant and parent with a large number of species in the community, perform this procedure, and end up with just an equation which describes to you what the mutant does. And the effect of the community is just some simple simple single parameter that suffices to capture it. And so you end up with an equation that looks like this. And really what I want to point out to you is this one additional parameter that we need. So just, the two parameters, which are the old parameters that we cared about, is S, the selection coefficient, how much faster does the parent go, the mutant grow compared to the parent. N, which is something called the effective population size, which tells you how much stochasticity matters. If you have larger populations, stochasticity matters less. If you have smaller populations, it matters more. And then really there's this one additional parameter, which we call eta, which captures what we call the ecological feedbacks due to all of the other species in the sort of community. And so really the lesson is that there's this one simple parameter family that captures these ecological dynamics. And you can ask now, does it actually make a difference? What are the consequences of including ecology compared to this classic Kimura picture, which just had the parent and the mutant? And the answer is there is an important consequence. And to see that consequence, what we will do is we will plot the classical curve that people care about, which is the fixation probability. To remind you, this tells you how frequently, how many trajectories the mutant will fix in the sort of population, as a function of this parameter S, the selection coefficient, how much the mutant is better than the parent. So if it's 0, the mutant and the parent grow equally well. If it's positive, the mutant grows better. If it's negative, the mutant grows worse. And so just to remind you, the Kimura formula, which you get by solving the Kimura equation, which is a no ecology formula, it looks, if you plot it, something like this. So like the fitter and fitter the mutant becomes, gradually the fixation probability goes up. And eventually, of course, if the mutant is very fit, you almost always fix. In the presence of ecology, with this model that I was showing you, you get a curve that looks like this blue curve over here. And just to sort of tell you in the last 30 seconds that I have, there are similarities and there are differences. And the differences are the important thing I want to point out. So of course, if the mutant is worse, of course, the mutant is not going to fix. These are deleterious mutations. But really, there's this important regime in red where the mutant is actually beneficial. It's actually better than the parent, but its fixation probability is significantly suppressed compared to the absence of the ecology. And of course, if the mutant is eventually very, very good, then of course there's no surprise that it'll almost always fix. So compared to this gradual picture, in the presence of ecology, there's an almost switch-like picture. Below some critical fitness, you don't fix. Above some critical fitness, you do fix. And it turns out that this critical fitness is related to these ecological feedbacks that I was talking about. So with that, I'll end. The takeaway that I want you to have for this talk is that despite the ecological complexity of communities, which might make you think that this is a hopeless task, actually the dynamics of mutants in these communities can be surprisingly simple and predictable. And this predictability, statistical physics tells us, is not despite, but because of the high diversity of the community. With that, thank you for your time. I'm happy to take questions.
Questions?
A wonderful talk.
Thanks.
So, did you try putting 2 or 3 different mutants?
Yeah, that's an excellent question. So there is this regime, this is a regime of population genetics, which is typically called strong selection weak mutation. Mutants come up one at a time. There's another regime, which is a regime of clonal interference, where many mutants come up at the same time. This theory will fail for that regime. And actually, yeah, it's pretty hard to solve things for that regime. So this is something that we're working on right now.
So when you're looking at a community, there are multiple species.
That's right.
And so you really need to deal with it as a vector. If there are P species,
that's right.
Are you looking at the P vector, or are you looking at the mutant species versus the rest pooled?
So you are looking at the community vector, but what you're doing essentially is you're figuring out a way to coarse grain that vector.
So you're looking at one particular species and pooling the remaining, right?
Exactly, because you care about the mutant. What has happened here.
But that's a major simplification, primarily because selection coefficients of the other species with which this species is interacting is also going to play a role.
Absolutely.
So you need to take S1, S2, so on up to SP, and that's a very hard problem.
Absolutely, yeah.
Yeah. So how do you actually validate this?
Wonderful question. So we actually did manage to validate this. So one of the predictions of this kind of theory is that this one, that the presence of the community will actually result in what is called negative frequency dependent selection. Communities will suppress the fixation probabilities of mutants. And so what we've been able to do is to look at data sets from the gut microbiome, where people have actually been sequencing very deeply, different isolates. And so people have been able to track frequency dynamics in them. And so by fitting those frequency dynamics to models of this kind, you can fit what these different parameters are. And one of our predictions is that this parameter eta, which captures the ecological feedback, will always have a positive sign, which is to say it'll always be negative frequency dependent selection. And that's indeed what we see. That's sort of prediction one that we've been able to validate. This was long time series, so you can make an independent measurement at every time for different patients. And so for all of these, we find that these eta measurements are consistent with a positive number. And the second prediction is that, what I didn't tell you is that this eta captures some coarse-grained properties of the community. And one of the properties that it captures is the diversity of the community. So what this predicts is that if there are sudden changes to the diversity of the community, you should actually change the strength of frequency-dependent selection. And so in the same data set, they had patients who went who went through like big antibiotic treatments that killed off big parts of their gut microbiome. And so what we were able to show is that with no access to that information, just looking at frequency dynamics, we were able to detect those time points reliably. So that's another prediction that we were able to validate.
Yeah, so here one question.
Sorry, I don't. Yeah.
So I mean, this term mutant which you have mentioned appears to be not so clear, because it has to relate to genotype or phenotype.
Absolutely, yeah.
Now, in a microbial population, there may be thousands of mutants.
Absolutely, yeah.
Some may have a positive value in the selection, some may not have, some have a negative value.
Correct, yeah.
So what do you actually mean by the mutant?
Yeah.
And I always feel that the mutation and selection, that goes hand in hand for the evolution. So in a microbial population, there are always thousands or tens of thousands of mutations.
Absolutely.
And what are you talking about? Which mutant you are talking about?
Yeah.
Can you please specify?
Okay, wonderful. So I heard 3 questions there, so I'm going to respond them in the order in which I remember them. So first was the question of what is exactly a mutant here and how do you reconcile it with genotype, phenotype. I think it's absolutely true that we don't have a good understanding of what genotype, phenotype maps are. So in the model, there's a very good sense that we have of what a mutant is. the model only deals with phenotypes of different strains. That's their ability to consume resources. And so in our model, when we have a mutant, what we have is we take a parent's phenotype, that is, it's some vector by which it's able to consume different resources, and we make a small mutation, which is to say we ask for a vector that's similar to this vector but not exactly the same. And so that exactly is it's a phenotypic mutation. What exactly this means in terms of genotypes, we don't know, but that's because we don't have a good understanding of exactly what the genotype-phenotype maps look like. That's answer one. Question two was about the fact that there are thousands of mutants in a community. That's, again, absolutely true and goes back to this question that was being asked. So it's true that many microbial populations are in the regime of clonal interference. And so, what we view this as is, these models didn't even have the community to begin with. And so this we see as sort of the first step towards going and describing the dynamics of clonal interference. So I completely agree that that's where we need to go, but we're not there yet. And then the third question was about, which mutant we're talking about here. I think that's an important question because one of the advantages and maybe disadvantages of an approach like this, which is theoretical, is that we are agnostic to a lot of the chemical details of what exactly we are talking about. So the advantages, I think, first I'll say the disadvantages. The disadvantages are that it's very hard to describe any particular system. If you want me to describe what is happening in the dynamics of your gut or my gut or the soil outside in the IISc faculty hall, I can't do that using this modeling approach. But the advantages are exactly that if you want to understand very generically and at a conceptual level what can be possible or what we typically expect, that's exactly what this approach is useful for. So I think the question that this is trying to answer is, typically what kinds of effects of mutants would you expect to see?
So this ecological feedback is a time-independent parameter?
Oh, great question. So typically it is time dependent. Yeah. But in this case, you get a remarkable simplification because of these two regimes of stochasticity dominated and selection dominated that I was telling you about. So it turns out that it's really only the early dynamics of the mutant that matters. And if for various reasons that I can describe offline. And so because it's the early dynamics that matter, you can build a very good approximation of and replace this time-dependent parameter with a time-independent one.
I was just thinking about viral evolution in the context of the immune system.
Absolutely.
And there are a lot of quasi-species, but their evolution is going to depend on the antibody response, which will be completely time-dependent.
So I don't think this will be a good model to describe, those kinds of dynamics.
Akshit here, very nice talk. A quick question. Is there any way to understand intuitively, perhaps I missed it, why the ecology always imposes a negative feedback?
Yeah, great question. So really the idea is that there are two kinds of ways that parents and mutants interact. One is, of course, directly, and the other is indirectly through all the different paths in sort of community. So it turns out that if you have these kinds of competitive interactions, you end up getting an enemy of my enemy is my friend effect. So you end up getting situations whereby, the mutant and the parent are kind of competing in the same niche. And so if the mutant is kind of doing better from the perspective of the community, this is just one niche. And so the parent does a little bit worse. So effectively, you kind of get this negative frequency dependent selection.
So last question.
Very interesting. But here we are saying mutation occurs in one species and doesn't affect other species. But in real life, mutation can jump from one species to another.
Oh, you mean through horizontal gene transfer and stuff? Absolutely.
Have you looked at that and how would that matter?
No, so these are, like I said, we view this as the first step. And these are absolutely the kinds of things that we want to play with in the future.
So your talk generated a lot of interest, you can see, but we have to close it. Thank you very much. I thank all the speakers and the audience. Thank you so much.
Okay, thank you very much, everybody, for being here for this meeting. I'd really like to thank the whole audience, to stick it out till the end and, attend all these interesting talks. I'd also like to thank once again all the speakers of all the sessions, whoever is here. and of course all the chairs who've spent a lot of time organizing it and of course taking care of the sessions. I'd also, I think, really need to thank the Academy staff because they are the ones who've put together the nitty-gritty of the meeting and, made it all very comfortable for us. So I particularly would like to thank them. There's an announcement. So lunch is being served at the IIC guest house, just like it was yesterday for everybody. But for the council members, we need to proceed for the meeting straight away. So lunch will be served at the Academy for the council members. So we need to just get into taxis and leave immediately. Okay. Thank you very much.